Plant and animal tissues. Tissues are groups of cells that aggregate together based on similar structure and function. Animals and plants are made up of different kinds of tissues. For example, groups of parenchyma cells form parenchyma tissues and muscle cells form muscle tissue. Different types of tissues have distinctive architectures that are well suited for what they do. Parenchyma and sclerenchyma tissues are found in plants, whereas striated muscle fibers and nerve tissues are found in animals. To identify parenchyma and sclerenchyma tissues in plants. Materials required. We will require permanent slides of parenchyma tissue and sclerenchyma tissue and a compound microscope. Procedure. Take the permanent slide of the parenchyma tissue and place it on the stage of the compound microscope. Adjust the mirror of the microscope carefully until the image you see has maximum clarity. Observe the permanent slide of the parenchyma tissue under the low power lens and then under the high power lens of the compound microscope. Move the slide very slowly back and forth to identify the different parts of the parenchyma tissue. Repeat the same procedure for the permanent slide of the sclerenchyma tissue. Observations. Parenchyma cells. The slide shows numerous cells. We can observe that cells are isodiametric, which means they have the same length and width. These are loosely packed cells, so large intercellular spaces are found in this tissue. Intercellular means between or among cells. The cells consist of peripheral cytoplasm and a prominent nucleus that is surrounded by a thin cell wall. Each cell has a large vacuole at the center. These cells help in photosynthesis, storage, and keep aquatic plants afloat. Sclerenchyma cells. We can observe that the sclerenchyma tissue consists of thick-walled dead cells. Sclerenchyma cells can be divided into two types, fibers and sclereids. We can observe that the sclerenchyma fibers are elongated and narrow with pointed ends. The adjacent fibers have oblique pits. There is very little or no protoplasm. The nucleus is absent in the cell. Vacuoles are empty and are called lumen. These cells provide mechanical strength to the plant. To identify striated muscle fibers and nerve cells in animals. Materials required. We will require permanent slides of striated muscle fibers, nerve cells, and a compound microscope. Procedure. Take the permanent slide of the striated muscle fibers and place it on the stage of the compound microscope. Observe the permanent slide of the striated muscle fibers under the low power lens and then under the high power lens of the compound microscope. Repeat the same procedure for the permanent slide of the nerve cells. Observations. Striated muscle fibers. The slide shows large number of very long and cylindrical fibers. The fibers are wide and non-tapering and enclosed within a membrane known as the sarcolemma. The striated muscle fibers are multinucleated, that is having more than one nucleus, and these nuclei are situated towards the periphery. Each fiber has many myofibrils embedded in the cytoplasm. Myofibrils are small, thread-like fibers that run through each muscle fiber. On these myofibrils are present characteristic alternate light and dark bands, giving these fibers its striated or striped appearance. Nerve cells. As observed, a nerve cell has a large body called cytin that contains a prominent nucleus. The cytin has cytoplasmic projections called dendrins. A dendrin is a short, branched, thread-like extension of a nerve cell which conducts impulses towards the cell body. The dendrin further branches out to form fibers of different lines called dendrites. One of the dendrins is long and is called the axon. The axons are covered with myelin sheath.